Guys, to in today's video we will be talking about cancer. Cancer is actually a very harmful disease, and there are several ways by which a person can get cancer. So actually let's see what is cancer. It is actually the abnormal and uncontrollable growth of human cells, or it could be any other organism cells. Any several organisms develop cancer, not only humans. So it's the abnormal and uncontrollable growth of cells. So let's see how cancer is there, how cancer can be caused. So let's see, there are two types of tumors. Before that, we need to know what is tumor. Tumor is actually a mass of undifferentiated differentiated cells which divide continuously, which may or may not be may not be harmful. Yes, the tumor could be harmful or it can't be harmful. So let's study the types of tumor here. Let me change the font. Types of tumors. So there are actually several types of tumor, but in this stage we will be studying about two main types, that are the benign tumor, and the second one is the malignant tumor. Both these are the tumors. So you know what is tumor, and now let's study their types. First we will be taking up the benign tumor. Change the color. Benign tumor are actually growth of cells which are undifferentiated. You can't differentiate between the cells. So let me make this histology. This is the cell. These are the normal cells, and yes, there is one tumor cell or the cancer cell. So this will grow continuously. This is undifferentiated. Before studying the tumors, first we should know which types of cell actually comes under the tumor cells. Condition for tumor cells. Let's see here. First, this cell must have lost its regulatory mechanism and should have lost the compact inhibition property of normal cells. So let's study what does regulatory mechanism means. regulatory mechanism means and what does compact inhibition means. So each and every cell, a human cell, this is the nucleus, this is the plasma membrane, each and every cell is controlled by the nucleus and several regulatory factors which are present in the cytosol of the cell. They actually regulate the whole metabolic process and the division process, the mitosis, the meiosis. They are actually under control of a defined regulatory mechanism which each and every cell needs to follow. And in order to follow these In order, the cells which follow this regulatory mechanism are the normal cells, whereas cancerous cells do not follow the regulatory mechanism, which a normal cell follows, which includes how it should be metabolized, how it should reproduce its progeny, and etc. etc. Whereas, let's study what is compact inhibition. So, see, there's a group of cells. I'm making a layer, and let's see, this is a cancerous cell or a tumor cells or a neoplasm. Yes, the tumor cells are also known as neoplasm. So this is a cancerous cell, and here these are the normal cells. So normal cells, when they divide, they grow extensively, and when they touch each other, the cell stops to divide further. Yes, after that, since there is no space here, the cell has been touched by the other cell. Now if it was a normal cell, it won't divide further, since it would release some chemicals which will stop the cell from dividing. But if this is a cancerous cell, if it is not following any regulatory mechanism, the cell will continue to divide and make an outgrowth, and this is what you called a tumor. Yes, so these two were the conditions for the tumor cell. So let's now study benign tumor. Let me change the font. Benign tumor. These tumors are actually localized, not harmful, slow growth. Do not cause cancer. As I told you, not every tumor is cancerous. Yes. So let's see how its mechanism works. So this is a cell, this is a cell, here's a tumor cell. So it will divide here, 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 and this will be a group of undifferentiated cells called tumor. But this cell will remain here only. For example, if this is a tumor in the epidermis of a skin, it will be localized there itself. It won't travel to the another part of the human body. Localized, it is usually not harmful. Yes, see, this is undifferentiated, and this is a huge mass of cell with respect to the normal cell, so it will also require huge nutrients, and so blood vessels which are actually supplying the nutrients here, the blood vessels will also develop here, which will transfer the normal cell nutrients to them. But since they have slow growth, this won't affect much. And also, these are non-cancerous. Yes, they are actually non-cancerous. So benign tumor, yes, you can have, and they can be easily removed, or you also can't remove since they are harmless. Most of them are harmless. In some of the cases they might be also harmful, but they don't cause cancer. So now let's see the another part, that is the Malignant tumor. Malignant tumor. Malignant tumor are actually the cancerous tumor. These are the cancerous tumor. They are they follow a process called metastasis. They also they are the cancer metastasis harm adjacent cell, may cause death. So let's see how it follows this mechanism. Let me make a group of cells first. Yeah. Let me see. This is the dermis. I'm making the epidermis. Dermis. And this is the epidermal cells, the epithelium cells actually. And here is the tumor cell and the normal cells. Now the tumor cells will start dividing here in the skin, and after time you would see a outgrowth of undifferentiated cell, which is the malignant tumor. So you would be asking, what's the difference here? Let me make the blood vessels here. These are the blood vessels from which the normal cells take the nutrients, and same here the blood vessels will develop to provide nutrients to the cancerous cells. And one point to note is that they grow fast as compared to the benign tumor. So since they grow too fast, they take up the nutrients of their adjacent cell, and the cell at the last may die off, which is not natural. It is unnatural because it's due to this tumor which is developing here. And also they follow metastasis. What is metastasis? It is the propagation of the tumor cell from the blood vessels or the lymphatic vessels from one place to the another, and causing cancer to different places in the human body from one place to another. Yes, now some segment of the malignant tumor would transfer to the blood vessel and would travel some far distance and would be released in some other part, and would cause cancerous growth in that part also. So yes, it's hugely harmful and may cause cancer in the several parts of the body. So yes, this is the tumor which is cancerous, and this the process which is followed is metastasis. It also harms the adjacent cell, as I told you, and may cause their death. As it grows fast, it requires much nutrient than the adjacent cell, and hence the normal cells are deprived of these nutrients. So yes, these were the two types of tumor. Now let us study what causes cancer. So for this, let us study the mechanism first. Let me make a cell. So this is a normal human cell. Let me make the font small. Here's the nucleus, and here's Chido. In every normal human cell, there is a part of DNA segment which contain the proto-oncogene or cellular oncogene, C-onc, as you can call it in short form, C-onc. But they are always present in inactive condition in a normal cell. Yes, you heard it right. So first see what does proto-onco means. As it's clear from the terminology itself, proto-oncogene. Proto, inactive. Onco means cancerous. Inactive cancerous gene. It is present in each and every human cell. But some substances or some agents or some physical agents may cause these genes to be active. Such substances or the process which leads to the to the formation of cancerous cells are known as carcinogens. Carcinogen. The substances which causes cancer are known as carcinogens. So these carcinogen may act to the proto-oncogene and convert them to the oncogene. Yes, and these oncogene may make the normal cell cancerous. So how do they do that? Okay, let me explain. This is a chromosome, typical chromosome in human cell, and the end part here, which covers the ending, are known as telomere. So when a normal cell divides, in each division the telomere gets broken and the chromosomal size decreases. The telomere always gets shorter, it always gets destroyed, and after a particular time the telomere is completely vanished, and it is that time when the cell stops dividing. The cell stops dividing. Yes, when the telomere is completely vanished from the chromosome, the cell stops dividing. There are certain enzymes which cause the stoppage of the cell division, and the cell is dead, which is the natural death, also known as apoptosis. Apoptosis, which is the natural death of the cells. So yes, this controls the unlimited division. So yes, you can say that human cells are immortal. They are not immortal like cancerous cell, but also here certain enzymes are present which are inhibitor of telomerase. I'm talking about the normal cell right now. Telomerase. You might be wondering what is telomerase? Inhibitor of telomerase. Telomerase are the enzymes or the group of proteins present in the cytoplasm of the cell which actually protects the telomere. You got it right. Now think, if there is telomere present, and imagine if there is no inhibitor of telomerase in the normal cell, the telomere will be persistent throughout the divisions. You got me? The telomere will be persistent during the division, and it will always be there. It will always be present, no matter how much divisions are there. So yes, the cell will be immortal. It will continue to divide since the telomere is always present there. Because I just told you that if the telomere is completely vanished from the chromosome, then only there are factors which stop the cell from dividing. But if there is no inhibitor of telomerase, the cell will continue dividing. But this is not the case. Inhibitor of telomerase inhibitor is always present in the normal cell. So in the cancerous cell, the when the carcinogenic agent acts to the normal DNA, they make the proto oncogene into oncogene, and this oncogene produces telomerase. It also stops the oncogene also stops the regulatory inhibitor, and the cell is now out of control and it will be dividing humongously. Now the telomerase is there. Also, when the tumor cells will be dividing, their telomere will not be losing, and hence there won't be certain chemicals which will be there to stop their division, and hence tumor cells are immortal. So this is what actually happens, which causes cancer. So let's talk about some of the carcinogenic agents. Yeah, carcinogenic agents. So there are physical agents which may include UV rays, yes, which may cause cancer. They are actually non-ionizing rays. They don't ionize. They are of less power. X-rays, gamma rays, they are ionizing. These all are the carcinogenic agent which may convert the proto-oncogene into the, yes, repeat with me, into the oncogene. X-rays and gamma ray. And also there can be physical. These were the physical part. Let's study the chemical part. Chemical agents, you might be knowing the soot which is there from the incomplete combustion of the hydrocarbons, which include the high high polycyclic benzene ring, aromatic ring carbon, which are carcinogenic. One of them is benzoprene, which causes skin as well as lung cancer. And yes, tobacco or smoking, this releases nicotine plus compound NN, I'm writing the IUPAC name, NN nitroso dimethyl amine. They cause, you must be knowing, lung cancer. Yes, you're right. Now let's move to the third chemical, vinyl chloride, which causes liver cancer. At the fourth one, there are certain elements: cadmium oxide, chromium, and nickel. This causes the lung cancer. Also, asbestos causes lung cancer. So see, these are some of them. Also mustard gas, which causes cancer. Let's see the biological agents. Biological agents actually include some of the viruses. Include viruses. This virus contains V oncogene, virus oncogene. Some of the virus contain virus oncogene. When they invade the normal cells, they release their V oncogene plus the P oncogene, proto oncogene or the C onc of the normal cell makes active oncogene. So they are also activators of proto-oncogene. They transfer their genetic viral oncogene into the human's normal cell and convert the proto-oncogene to the oncogene. Now this oncogene would cause cancer. Yes. So these were some of the carcinogens. Now let's see the diagnosis method. How you can get to know you are having cancer. My bad. First is the, let me remember it, biopsy. Yes, it's the biopsy, which is actually a histopathological method. Histopathological method. In this, the part which is actually affected by cancer is taken, the sample is taken and it is stained, and it is told whether you are having cancer or not. It is actually taking the part of the cancer cell. Second include radiography. This may include X-ray, CT scan and MRI. In X-ray there is mammography, which is actually a way of knowing about the mammary cancer. The CT scan actually uses the X-rays only, and they give a 3D view of the organ affected, and the full form is compute tomography. And the MRI, which is the safest of all, it uses magnetic fields which is passed through the effective tissue, magnetic resonance imaging, and helps to diagnose the cancer. So this was the radiography. Third is the antigen testing. So if you're having cancer cells which are actually abnormal, your immune system actually want to be calm, they would be producing some of the antibodies. So yes, antigen testing is there, and there is a particular antibody, herpesine, which is released in the mammary cancer. This is an antibody, and if there is antibody, actually there is an antigen. So this was the antigen testing, this was the radiography, and this was the biopsy method. And now there is a molecular technology, biomolecular technology. Which helps us to actually see the part, the gene of the cell, the tumor cell, and help us to know which type of cancer is there. It works on the molecular level. So these were the ways to diagnosis. So now let's see what are the types of cancer. Types of cancer. So broadly speaking, there are three types: carcinoma, 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 sarcoma, leukemia, or the lymphoma. Carcinoma, carcinoma, carcinoma are the cancers in the epithelial cells which are ectoendodermal in origin. This includes, example, lung cancer, colon cancer, breast cancer, and several uh, gland cancer. Sarcoma virus, this would be mesoderm, and the connective tissues. Example, bone, cartilage. Thank you.